variety of major medical illnesses that can be chronic and can also be fatal, like heart disease, but also type 2 diabetes and cancer. Here in the UK, Cancer UK has started a advertisement campaign where you'll see these ads at bus stops and tube stations and things like that, where they're trying to make the public aware that obesity also predisposes to cancer. I think that's something that quite a lot of people aren't aware of. So the reason why this is concerning is because of the prevalence of obesity, and the prevalence of obesity keeps going up. And in the United Kingdom, as well as in the United States, more than half of people are overweight or obese. And what you can see here is a map of how obesity is spreading across the globe. And you can see the darker sort of orange, the color, the higher the rate. And so it's not just a problem that the UK has or the United States has, it's actually all over the world. And one of the biggest concerns is that childhood obesity is increasing at a faster rate. And the problem with this, as I just said, is that there are all these chronic conditions that are associated with obesity. So you can imagine that if children are developing overweight and obesity at an earlier age, then this is predisposing them to medical problems in the future. And so that's really, that's the reason why I'm interested in it, why I'm interested in researching obesity, is because I see it as a concern to human health. And when I mentioned, when I was doing my PhD, how I was noticing the change around me, some of you may have seen these slides already, but they were put together by the CDC. And it's just for many of you who might be watching this, you weren't born in 1985, so you don't realize that not that many years ago, actually, overweight and obesity were very uncommon. A very small percentage of the population was overweight or obese. And in fact, some of the early advertisements were how to gain weight rather than how to lose weight. So this is 1985, and what you can see is how the landscape is changing just year on year, and how the rate of overweight and obesity is increasing. Now, my family is from the South, so you can see the South is where some fried food, etc., is a little more prevalent. They're changing faster than some of the other states that are more associated with exercise, like Colorado. And so you can just see how rapidly the landscape of the United States changed. So drilling it down, how is this happening? So, the environment's changing. So, and the food is changing, but, so what's happening within our bodies? How can we influence obesity? And I like to sort of divide it up into three major ways to think about it. One is energy in, one is energy out, and one is energy storage. And in order for body weight to be stable, we need the calories that we're taking in to equal what our body uses every day, so our energy expenditure. And the current medications that are available are, some of them tackle energy storage, like orlistat or Ali, and what that does is it inhibits dietary fat from being absorbed. There's a lot of interest and excitement around targeting energy expenditure. So, physical activity is something we know about, but also trying to find ways to burn fat without exercise. And that might be something that would be appealing on hot days like today. But historically, most of the medications have focused the energy in component of the energy balance equation. And in particular, most of the medications have looked to the brain to try and control appetite and energy intake. And what I'm going to talk about today is 5-HT or serotonin and how it links in with the melanocortin circuits in the brain. When serotonin was discovered back in the 1940s by Robert Greene and Page, this is not something that they were thinking about. They weren't thinking about food intake yet. When they found serotonin, they found it in the serum, and they found that it caused constriction, and so they named it serotonin. And in the 1970s, what researchers realized is that there's an inverse relationship between brain 5-HT and food intake. So the more 5-HT that is released, the more the bigger the reduction in food intake, and vice versa. And so this is where I come in. This is supposed to be me. So when I was doing my PhD, one of the questions that I asked is, okay, there are a lot of medications on the market or used in the clinic to influence serotonin. So SSRIs like Prozac, for example. So when we think about Prozac, we typically would think about a drug that is used to treat depression or anxiety. But I was wondering if it's increasing the bioavailability of serotonin, of our own body's serotonin, could this improve obesity? And I should also just mention, I should have said on the previous slide that that serotonin is also called 5-HT, and so I will try and stick with one or the other, but I do kind of mix it around. So can 5-HT medications improve obesity? So what I did for my PhD at Tufts is I gave rats Prozac every day for a month and looked at whether or not that affected food intake and body weight gain. And what I found is that actually, yeah, it worked really well at reducing food intake and body weight. And sort of a similar approach was also being developed and released in the clinic at the same time, which was difenfluramine, which was considered to be a revolutionary new weight loss drug. And what difenfluramine does is it blocks the reuptake of serotonin and stimulates its release. But shortly thereafter, reports were released that difenfluramine was associated with valvular problems, and it was removed from the clinic. So the challenge then became, can we dissociate this therapeutic benefit from the side effects? And so we already knew from the human, from patients, that 5-HT does reduce food intake and body weight in people. So, where is serotonin produced? Where is it acting to produce these side effects and therapeutic effects? And as I mentioned before, it's quite handy that the researchers in the 1970s had already figured out that it was brain serotonin that was affecting food intake and body weight. And the way that they did that is they knocked down, they sort of prevented the release and production of serotonin just in the brain, and rats became hyperphagic and obese and developed diabetes, type 2 diabetes. So looking in the brain, where is serotonin produced? What's produced in 9 different locations that early anatomists labeled B1 to B9, and you can see them here in blue. The biggest source of serotonin made in the brain is in the dorsal raphe. So that seems like, the first place to look. And we looked at various different serotonin circuits in the brain. And we also looked to characterize these serotonergic neurons in the dorsal raphe. And the dorsal raphe, as I said, since it's so large, it actually can be thought of as multiple structures, not just one homogeneous structure. And this is something that we began sort of characterizing. Well, what are these serotonin neurons that might be involved in food intake? You know, what kind of receptors do they express? Where are they projecting? What kind of jobs might they be responsible for? And we found that the serotonin neurons in the dorsal raphe don't express leptin receptors, but do express other receptors that could be involved in appetite regulation. And we were looking at, these projections as well. And one of the projections that we were interested in was the serotonin projecting to the amygdala. And what we found with Tom Cash's lab, and when I was doing my postdoc with Larry Teacott at UCSF, is that serotonin here, this sort of amygdala circuit appears to be more involved in anxiety. And we also looked at the ventral tegmental area with Yang Yu's lab. And Yang Yu also did a postdoc with Joel Elmquist like I did. And that's how we got connected. And what we found is that serotonin projecting to the ventral tegmental area is more involved with reward, motivation, and binge eating. So then we were thinking, okay, well, where's a likely target where it might be acting to affect homeostatic feeding? And the arcuate nucleus seemed like a logical choice. So we had, we've been investigating that circuit. And just to give you a sneak preview, we've got a paper that was led by Yang Yu's lab that will be published. I'm guessing it's just been accepted in molecular psychiatry, and I'm guessing that it will be published either later this month or next month, so have a look out for that one. And the reason why we were interested in the arcuate nucleus of the hypothalamus is because it's a really important homeostatic brain region. And in the arcuate nucleus, we have, which I'm sure Steve O'Reilly, if you spoke with him recently, will have talked to you about the melanocortin circuits. And what's really cool about the melanocortin circuit is that we make our own agonist and antagonist or inverse agonist. And these neurons sit side by side in the arcuate nucleus. And one acts to increase hunger and the other one acts to decrease hunger. And we know this from pharmacology studies and genetic studies. And they compete for action at the melanocortin 3 and 4 receptor in the brain, with the melanocortin 4 receptor being the one that's really taking the lead in the regulation of food intake and body weight via POMC and AGRP. And so work that we did at Beth Israel with Joel Elmquist, one of the things that we looked at is how is difenfluramine. We know, as I said, we know that this drug works. It's been prescribed globally. It's reducing food intake and body weight in humans. How is it working? We had this hypothesis that it's working through POMC neurons, through these melanocortin neurons, and I'm not going to go through all the data, but we ended up getting support for that. And so, so in asking the question, how are 5-HT medications like the difenfluramine improving obesity via regulating the activity of POMC neurons? So how is this happening? Through which receptor? And here we would turn our attention to looking at the 5-HT receptors. It's a little complicated because there are multiple 5-HT receptors, and what they've been organized and reorganized again. So when you're reading papers about the serotonin receptors, just be aware that they've been renamed quite a few times. And actually, we recently published a review paper in the Journal of Neuroendocrinology where I've actually listed a table of how they were renamed and over the different decades. But currently they're clustered into 7 different families based on sequence homology and effector pathways. And through this work, it's been identified that the serotonin 2C receptors, which used to be called the serotonin 1C receptor, but they've been renamed the 2C receptor, is the one that's the most important in serotonin's control of food intake and body weight. And one of the ways that we know this, and this paper was published by my postdoc supervisor. This is what a mouse line that he generated. We know from knockouts that pretty much all the serotonin receptors have been knocked out, and the mouse that leads to a hyperphagia, as you can see here, and obesity phenotype is the serotonin 2C receptor. So based on pharmacology and genetics, we've been able to identify the serotonin receptor that is responsible for most of serotonin's effects on food intake and appetite, food intake and body weight, and that's the serotonin 2C receptor. And the clinical significance of this was realized with the release of the serotonin 2C receptor agonist, lorcaserin. And it was the first new drug that was approved for obesity treatment in the USA for around 12 years. It then was, last year, it was voluntarily withdrawn from the US market for obesity treatment. So the question is, again, how is it working? So we're thinking that the way that it's working, this is the proposed model, is we know that diphenfluramine is using the melanocortin circuit to produce its effects on feeding. And so the idea is that lorcaserin does the same sort of thing, that it's coming in and acting at the serotonin 2C receptors on POMC neurons. So here's me asking the same question. Does lorcaserin also require the POMC pathway? And this is just a schematic just illustrating what I've just said. So here lorcaserin would be acting directly at the serotonin 2C receptors, influencing the activity of POMC neurons to stimulate the release of the POMC peptides, to act at the melanocortin 4 receptors, to affect food intake and body weight. And one way that we had to look at this is we took advantage of a mouse line that Malcolm Lowe created, where POMC was only knocked out in the brain. And as you can see here, the wild-type mice, as you would expect, are responding to lorcaserin by reducing their food intake, whereas the mice that don't make POMC, so the brain POMC knockouts, are not responding to the drug. So what that tells us is that POMC is necessary for lorcaserin to affect food intake and body weight. Now, POMC is made in 2 different places in the brain. It's made in the arcuate nucleus, and it's made in the nucleus of the solitary tract. And this, I'm just going to throw out a little side note here. For people who are using the POMC Cree line for different studies on things, you might see POMC expression in other brain regions. But in the adult, POMC is only produced in the arcuate nucleus and the nucleus of the solitary tract. So the other cells that you might be seeing as POMC positive, they're not fated to be POMC. So they're not POMC in the adult. So it's just the 2 places. We have 2 places that we can investigate, the arcuate nucleus and the nucleus of the solitary tract. And both of those are really important homeostatic brain regions. And as you can see in this schematic, they're sort of surveying, they're sort of getting signals from a variety of different places in the periphery that are important for body weight regulation. So they're both really well positioned to be getting information about energy availability. So which subset of brain POMC neurons does lorcaserin require. And to investigate this, we looked at both of them, both populations, the arcuate and the NTS. And what we found is that in both the arcuate and the NTS, about 40% of the POMC neurons contained the serotonin 2C receptors. So both subsets of POMC neurons have the capability to respond to serotonin via the serotonin 2C receptors. We then, in collaboration with Michael Cowley and also Dave Lyons, did some, Dave Lyons was a postdoc in my lab, and he did the NTS work. We then looked at whether or not the neurons are responding to 5-HT2C receptor agonists and lorcaserin, and we found that they were. So if you apply a 5-HT2C receptor agonist or 5-HT itself, the POMC neurons depolarize. So they're activated by 2C receptor agonists. So then to try and understand whether or not POMC was required for lorcaserin's effects on feeding, a very talented postdoc in my lab who now has his own lab, Pepe D'Agostino, he's now at the University of Manchester. What we did is we knocked down POMC specifically in the arcuate and specifically in the NTS, and then we looked at feeding. And what we found is that POMC neurons in both regions were required, but they were required for different time frames of lorcaserin's effects on feeding, with POMC in the arcuate being required for lorcaserin's longer term effect on feeding, with POMC in the NTS being responsible for the shorter term effect, so the more immediate effect of lorcaserin on feeding behavior. So this is just to summarize. So what I've told you already is that drugs like diphenfluramine are require POMC neurons, and that so does lorcaserin, and that that what serotonin does, and also what the serotonin 2C receptor agonists do, is they activate POMC neurons, which then require downstream melanocortin 4 receptors. I haven't shown you those data, but they're in some of our papers. So the next question that I had, and this was originally funded by the American Diabetes Association and also the NIDDK, is whether or not the serotonin 2C receptor medications could improve type 2 diabetes. And the answer would be, you would expect, would be yes, because if you think about reducing food intake and body weight, if you're looking at a sequential model, you should see weight loss and then improved insulin sensitivity. So it should be yes. But the question that I had is, could it have direct effects or independent effects on insulin sensitivity? And at this time, this was a pretty strange idea, using the brain to influence insulin sensitivity, because all the current frontline medications now act in the periphery at the specific tissues to produce the effects on insulin sensitivity and glycemic control. So that why would our bodies, why would we have that sort of mechanism of going to the brain to control insulin sensitivity? It was kind of, it seemed like a strange idea, but luckily the American Diabetes Association and the NIDDK were willing to take a punt on a strange idea. So that's also just a point to say that if you have a kind of strange idea, don't be talked out of it, give it a go. You never know, it might be right. So when we did this, what we found is that if you give lorcaserin to mice that have diet-induced obesity, straight away, immediately, you're affecting, so no time to change food intake, no time to change body weight. You're seeing an immediate effect and dose-related effect on glucose and insulin tolerance. So it does have an effect. And we were interested to see whether or not it also requires POMC neurons. So we know that POMC is important for lorcaserin's effects on food intake. Is this also how lorcaserin is affecting glycemic control? And so we again used the POMC knockout mice. And what we found is that in control mice, the 2C mice or wild type mice, they responded as expected to lorcaserin, but if mice didn't make POMC, then the mice didn't respond to the drug. So how is it working? We weren't really sure. Is it stimulating insulin secretion? No, it's not doing that. Is it affecting hepatic glucose production? Yes, it is. That's what we found, is that it's one of the ways that it's working is it's affecting hepatic glucose production. And with Mark Evans, we did clamp studies in mice and to look more specifically at how lorcaserin is improving glucose homeostasis. And what we found is that, again, it's affecting hepatic glucose production and insulin sensitivity. And with Justin Rochford, we looked at, okay, which tissues, where is it improving insulin action? And we looked at the liver, muscle, and fat, and we found that it's affecting insulin binding to its receptor. It's improving insulin sensitivity in the liver and muscle. And if given over a 1 or 2 week period, what we're seeing is a reduction in the enzymes used for glucose production, PEPCK and G6P. Also, when we gave it over a 2 week period, this is now just a 2C agonist MCPP, what we found is, again, we're making sure that we're not using a dose of the drug that is affecting food intake or body weight. In order to do that, we need to go for a higher dose of the drug. But for this lower dose of the drug that doesn't affect food intake or body weight, we're seeing a reduction in hyperinsulinemia. So this is just a little movie showing you how it works. Taking lorcaserin then signals to the brain, in particular to POMC neurons, which then signal to the liver to reduce hepatic glucose production and also to increase insulin sensitivity, and also in the muscle. And what's important about this is that this research that we did in mice has been replicated in patients. So there have been quite a few papers out on this and showing that lorcaserin improves diabetes. It also reduces the transition of prediabetes to diabetes, and it also boosts the efficacy of metformin. So it actually is working to improve diabetes in humans as well. So this is the last slide, or the second to last slide. This is the summary slide. And just to say that what I told you about today is how lorcaserin is acting in the brain via POMC neurons, and we know this because we've turned POMC on and off. We've activated POMC and we've turned POMC off genetically. And what we found is that it is acting through POMC neurons to affect insulin sensitivity and body weight. And it's doing this via different circuits. So it's at the melanocortin 4 receptors where this divergence occurs. So it's affecting POMC neurons in the arcuate and the nucleus in the solitary tract for feeding. And then the melanocortin 4 receptors are different for the circuit. And I didn't tell you about that today, just in the interest of time, but you can look up some of our papers and read them if you want to read more about it. And of course, none of this work could have been done without the talented people that I am so fortunate to work with. I've had some just outstanding postdocs and outstanding supervisors. So I've just been really, really lucky, and some, fantastic funding as well that has helped this. Just great mentors and great people in the lab. So just been really fortunate. So thank you very much for your attention, and I am happy to take questions.
Thank you very much, Professor Heisler. That was really, really fantastic talk, and
Thank you.
took us through a long
journey.
Yeah, like it's really an enterprise of figuring these things out over an extended time. So we appreciate the kind of big picture view of that. So I'll kick it off with a question. So one thing I always wonder about when I think about serotonin and the control of appetite is like, what is known, or what do you think is the role of serotonin of this dorsal raphe arcuate circuit in normal physiology? Like, is anything known about what regulates serotonin release in respect to the control of body weight? Or,
yeah, it's a good question, and don't have an answer to it at the moment. I think one of the things, it's really interesting in thinking about that. I think POMC neurons are really important in regulating POMC and AGRP. The sort of other side of the coin are really important in regulating appetite and body weight. And they express receptors from a variety of different factors that are really important in affecting food intake and body weight, like, for example, leptin receptors, etc. And so what's interesting about it is that I think what happens with serotonin, whereas you can get leptin resistance, if where the cells aren't responding as well to leptin, for example, or not responding as well to insulin, for example, but they still seem to respond to serotonin. And so that's why the serotonergic drugs have been successful in reducing food intake and body weight, whereas the other things like leptin weren't successful. Now we're looking at ways to improve leptin sensitivity as a different strategy. So it's an interesting question. So what is serotonin doing in normal physiology? Presumably it's also going through that circuit. So as I said, as our brain as serotonin levels are fluctuating, you see increases and decreases in food intake. So I guess it's just another one of the inputs to let us know about energy availability.
Great. So we have a question here from Felix Schmittner, who asks, did you already investigate how stress or cortisol medications can affect these pathways?
Yeah, it's a good question. Because, as I mentioned, serotonin is involved in stress. It's involved in, through the 2C receptor. It's involved in HPA axis regulation. It's involved in anxiety. So, how are these things linking together? Certainly in fight or flight, you don't want to be hungry if you need to escape. So, maybe there's a link in that way. POMC is involved in the production of ACTH and then cortisol. So, these are, there's so much exciting work to be done. So I just encourage you to take that one and run with it. It's, there's a lot there to work with, and how these circuits all interact is something that's yet to be defined.
Yeah. Another one here from Hassan Mohammed, who says, great talk. How does lorcaserin affect anxiety or depression? Does it act on the same circuitries that regulate body weight and emotions?
Yeah, so it's yeah. I mean, I think again, it's all going to end up being sort of dose related. I don't know that serotonin's involvement in depression seems to be more related to the 1A receptors, not the 2C receptors. But certainly the 2C receptors are involved in anxiety, but there will be other receptors that'll be involved as well. But the 2C receptor is an important one in anxiety. So, it's there. I think there's going to be a dose related factor there. So lorcaserin doesn't affect anxiety. But like I was showing you before, what we're seeing in mice is we're seeing the first effects seem to be on glycemic control and insulin sensitivity. Then we see an effect on food intake. Maybe then you'd see an effect on anxiety. So the human in the patient studies and the patients taking lorcaserin, the biggest side effect was headaches, which is pretty much same for everything.
Mm-hmm. So one other question here from Rita who says, really interesting talk and historic perspective. I was wondering, is there an effect of lorcaserin by decreasing glucose production on either muscle or liver metabolic or functional homeostasis?
Yeah, I don't know. I mean, this is something that, it's what I don't know is exactly how it's working. How is that signal getting down to the, so that would be via POMC. So I think it's going through the autonomic nervous system. So you've got POMC neurons that are projecting to melanocortin 4 receptor expressing neurons in these pre-autonomic sites in the brainstem as well as in the spinal cord, which is then relaying the information to the organs. So beyond that, how it's all working, I don't know. It's still yet to be discovered. So again, that's another area that's just wide open to try and figure it out.
Interesting question here from Lidawi. Sorry if I pronounced that wrong. Thanks for the nice talk. Do you know if lorcaserin could also be used to treat gestational diabetes, or would it disrupt fetal brain development?
Yeah, again, really don't know. We have looked at some other types of diabetes and have found that, and I don't think we've published it yet, so I probably can't say it, but not gestational diabetes, but we've looked at other types of other models of diabetes and found that it also works. So, so quite possibly it could work there as well. But again, a whole nother area to explore. And, would it affect brain development? You'd have to do the studies to find out. The serotonin 2C receptor, I mean, serotonin is a trophic factor as well. And so, yeah, I mean, you just have to do the studies and have a look.
Yeah. Great. Well, oh, hang on. Just had another question pop up. So this one from Vort. Great presentation. Thank you. Do you know if serotonin mainly affects homeostatic or more hedonic hunger in humans, i.e. homeostatic or reducing motivation to eat food?
Right. So that's something that I think from my perspective, just looking at how serotonin is, how it affects homeostatic as well as hedonic feeding, I just would have expected that it would have worked better. But it does do both things. And the question is, but through different brain regions. So I think it's doing both. In humans, what we're seeing, I think the end result either way is what we're seeing is a reduction in food intake. So when we looked in mice, we looked at, and this is a paper that was published in Neuropsychopharmacology. We looked at home cage feeding using the same doses that we were using in, in the operant chambers as well. And so what we found is that the same doses were producing, effects on home cage feeding, as well as responding for food rewards like chocolate and yummy things like sugar. So, so it seems to be doing both.
Yeah.
In mice. So I would guess it would be the same in humans. It would be doing both.
Right. Great. Interesting questions. Thanks everyone for asking them.
Yeah, really good
we have
questions.
one more. Since we have some time, we have one more. So I'll ask this one. This is from KTB, who says, could you clarify the SSRI dosages that have found the results you mentioned in humans? Do you think there is potential for this medication to affect the obesity-related factors you have investigated and depression or anxiety simultaneously?
Right. Short answer, no. So what happened was, so SSRIs are affecting serotonin bioavailability everywhere. And so most of serotonin is not found in the brain. Most of serotonin, 98% of it, is not found in the brain. It's not made in the brain. And it doesn't cross the blood-brain barrier. And so, so when you take a drug like an SSRI, you could be affecting serotonin activity at all the different receptors that are expressed all over the place. And so that's what happened is when a drug like which blocks the reuptake of serotonin and stimulates serotonin release, when it was given to obese people, it worked really well at improving obesity, but it also then caused side effects. And so so I think that you wouldn't recommend that it be used as an obesity, that SSRIs be used as an obesity medication for that reason. And that's why, my next question was, okay, let's refine it down to the receptor that's mediating the therapeutic effect. With regards to people taking SSRIs for other reasons, like anxiety or depression, way that I think about it is that if that drug is working to improve anxiety or depression, what it suggests to me is that the serotonin system might be, serotonin levels might be altered in those people. And that's why taking a medication that increases the bioavailability of serotonin sort of is restoring or affecting the serotonin levels. And so if you think about it that way, correcting or restoring serotonin levels, you're not getting more serotonin, you're just getting, you're bringing them to more like typical levels. This is just the way I think about it. So, this hasn't really been done. This hasn't, but if you look at the clinical data, what you find is that some people who take SSRIs, some people who are depressed eat more, and some people who are depressed eat less. And some people who then take an SSRI, sometimes they will eat more. The people who ate less might then eat more. Some people might eat less. It's, then some people might have no change in food intake. So it's all over the place. When you have a human or somebody who doesn't have anxiety or depression, and you give them a drug that increases serotonin bioavailability, then you see a decrease in food intake. So it's that inverse relationship that I told you was established in the 1970s. So it depends on what your baseline level is, and as to whether or not that would work. But that's why the SSRIs affecting other receptors in other tissues like the heart is the reason why the drug discovery went into refining it to a specific receptor target so that you didn't have the side effects. You don't see these side effects. I should also just say you don't see these side effects in other people. So, you know, SSRIs are widely prescribed and you don't see these. It was specifically obese people where you see the side effect.
Great. Yeah. So, as I was saying, great questions. I think it really started some interesting.
Lots to think about. Yeah, lots to think about. I think that, you know, you guys are asking questions that are just like, they're so next step, you know, figuring out, you know, what's the, you know, how does this work? How does that work? How does that work? You know, how does it, it's just, these are fantastic questions and great, you know, next step areas for research. And also, you know, looking again, going back to the SSRIs and looking at how they're working and in different conditions and things like that. Really great questions.
Yeah, and if for anyone who's interested in continuing the discussion and maybe going into some more details, feel free to join the Meet the Speaker session, and the link is right there in the chat. So we'll meet there in like 2-3 minutes. And I hope to see many of you there. For everyone else, this is our last session of the season, so we'll be back in I think mid-September. So have a great summer if you're in the northern hemisphere, and we'll see you in September. And thank you very much again, Professor Heisler, for joining us and for a fantastic
Bye, everybody.
talk. Bye.
Thank you.